So our first unit that we're going to start with today is a scientific method. And so, first day of school, I asked you, what is science? Is it a bunch of things we know, or is it a way to learn new things? And it's both. This unit is focused on, okay, how do we learn these new things? What do scientists do to gain new information, to figure out new things? That's what we're talking about here. And so, excuse me. Usually in our notes we have like a bunch of definitions. They're just kind of for your reference. So if you need to know a definition of a word that we're using, go back and look at that. So let's look at an example to start us off. A student wanted to study the effects of red and blue light on plant growth. And so she set up an experiment. She set it up with the conditions shown at the top of that table. And then at the end of the experiment, plant 1, this one on the left, had grown 10 centimeters. Plant 2 on the right had grown 5 centimeters. So she set up her experiment, she looked at that data, and she came up with a conclusion. Her conclusion was, plants grow better under red light. Does anyone want to comment on her conclusion? Is that correct? Is that a good conclusion? Is it valid? Mateo, what do you think?
It's not valid because she changed everything. So
Okay.
you're supposed to just change one thing.
Okay. What do you mean she changed everything? Whether
Plant 1 had 2 weeks, plant 2 had 1 week, plant 1 had 1 liter of water, plant 2 had 0.5 liters of water,
yeah.
and then 15 Celsius and 20 Celsius.
Yeah, so there are many factors. So we have a word for all of those things. You know the word we use for all those different factors that Mateo is just enumerating there? Richard? No, not quite. Those different aspects, each of these things, okay, we call those, what, Sandy?
Variables?
No, what'd you say?
Variables?
Yes, variables. Those are all variables. So you guys know something about variables, right? Mateo just mentioned it. So in this experiment, does she actually know why plant 1 was taller?
No.
No. She can't be sure because she had a bunch of differences. So how do we improve upon this experiment? The word we would say is, how do we make this a more valid, that's the word we often use, valid, experiment? Talia?
You should have the same temperature, same like height of water, and then same time and same plant.
Okay. Not the same light?
No, because the experiment was which light makes the plant go red.
Okay. What do you guys think? Good. You're right. So yeah, the way we would properly set up this experiment is we'd have red light and blue light, and then we would try to keep every other variable the same. Doesn't really matter what we chose, whether we chose 20 or 15 degrees Celsius or 1 liter, half liter of water, but we just have to be sure that everything is the same. You guys ever learned the term, the independent, dependent variables? Is that something you guys, terms that you heard in elementary school or no? No? Okay. Did you learn this concept that you should have only one thing different between your groups? Okay. Yeah, so this would be a much better experiment. If she had set it up this way and ended up with the same result, then could she be more sure of her conclusion? Yeah. But in the way she originally set up this experiment was incorrect. Okay, so you guys have an idea about experiments, right? So we'll talk more about this and the names for these variables and so forth. But for now, let's talk about the steps of the scientific method. Now, we use 6 steps in this order. Different people might talk about the steps in different order or use different terms or a different number. There's not really a set, this is the scientific method. We do this, then we do this, then we do this. We have general principles we use to set up a good experiment and understand the data that we collect. But you can do it in lots of different ways. For our purposes, we're going to say there are 6 steps in the scientific method that you're going to have to know. You're going to have to know the names of them in order and what they mean. So to help you to do that, to remember them, I have a little sentence here. Sentence is, people really hate elephants on cars. If you can remember that exact sentence, you'll be able to remember the name of each step in the scientific method. So remember the sentence, or if you look at this picture, it will help you, this picture of Mr. Akiri standing on the car. If you remember that picture, then that will help you to know the steps. Each letter stands for one of the steps of the scientific method. So the P. Anyone have any idea what the first step might be? This one sometimes you might think you know, but maybe not. Dan?
Problem?
Yeah, it's the problem step. Is that what you guys are going to say? Okay, the problem. I'm not going to explain them really right now. I just want to list them. Anyone know what the R might stand for? Research. Have you guys learned these steps? Oh, you did? Everybody? Oh, okay, no problem. Then what's the H stand for?
Hypothesis.
Wow, okay. Everybody, the E.
Experiment.
It wasn't very good synchronization. You look at your conductor. How about the O would be?
Orthodox. I don't know.
Obviously, Dan. It's okay. Good thing our brains can grow. What about the O? Does anyone else have another idea? Jeremy? Observation. And finally, the last C stands for?
Conclusion.
Conclusion. All right. So what do all these words mean? That's what we'll talk about next. So problem, research, hypothesis, experiment, observation, conclusion. Those are the steps you'll have to know. People really hate elephants on cars.
I don't blame them.
You don't want that thing stepping on your car.
Oh.
Especially if you're driving a car that small.
Oh my god.
Or the elephant is that big. Okay, so in our experiment, next page. In our experiment, we start with a problem. Now, in the scientific method, we're talking about the problem. It's the question we're trying to ask. It's what we are trying to figure out in our experiment. So you always phrase the problem as a question. So in the experiment we just read about, the girl was asking a question, do plants grow better under red or blue light? That was her problem. And you could ask all sorts of different questions and answer them using scientific method. Then after you have the question you're asking, you probably need some background information. We call that the research step. You know, like, before you can set up an experiment or really even come up with your hypothesis, you need to know, okay, a little bit about the background of these, the subjects you're studying. In that plant experiment, what do you think, what would the girl have to know? What are some information she might be gathering before going forward? Yeah, like how much water do you need to give these plants? What kind of potting soil should I use? Can I grow lilies inside or whatever? So there's a whole bunch of little things you probably need to figure out before you can actually go about conducting your experiment. That's the research step. That brings you to the hypothesis. Now, when I say what is a hypothesis, you are all going to say a hypothesis is?
An educated guess.
Yes. Well, not wrong, not great. So, an educated guess about what?
Science.
Stuff, science. That's why that's not a great answer, because I get answers like stuff. All right, so it's more than an educated guess. Really, in your hypothesis, it's a prediction about something specific. Your hypothesis is your prediction Not about the weather tomorrow, but about what?
Your conclusion?
About, yeah, what is going to happen in your experiment. You predict what's going to happen in your experiment before you ever even do it. That's what your hypothesis is. It's a prediction about what will take place in your experiment. A good hypothesis is often written as an if-then statement. If plants are grown under red light, then they will grow taller. That's a prediction. And so once you make a prediction, how are you going to know if your prediction was correct or not? You do an experiment. The next step. An experiment's purpose is to test your hypothesis. It's a procedure you set up to see, was my prediction correct or incorrect? Experiments test hypotheses. As your experiment is progressing, you make observations. Observations are collecting the data from your experiment, measuring things. What were the observations the girl made in the experiment we just talked about? What observations did she make at the end of her experiment? Christopher? Well, you're getting a little bit ahead of me. How did she know that? What did she observe? Yeah, the height of the plant. Those are the observations she made. What did you just give me? You're giving me the conclusion. Because once you have the observations, the data, you look at it and you say, okay, now that I have data, was my hypothesis, my prediction, correct or incorrect? Is my hypothesis supported or not? That's the last step, the conclusion step. All right, so let's think of another example and apply these steps. Now that we know what they are, let's apply them to an example. Does anyone have pet mice at home?
No.
Hamsters?
No.
Rats?
No.
Gerbils?
No.
Guinea pigs?
No.
All right, well anyway, let's pretend that you did. And we want to do an experiment. We want to know, so we're going to ask a question. Bless you. Do mice gain weight if they are given extra vitamin C? That's a question. So we call that the problem step. Can we answer that question in an experiment?
Yes.
Can you like envision a way we might test that or figure that out?
They give vitamin C.
Yeah, we'll get there. All right, so let's say we're gonna try to do that. We aren't really gonna do it in class. What research? What might you have to know before you can move along in this experiment? You need to have some background knowledge. Drew, what's something you'd have to know?
How much vitamin C a rat usually gets?
Yeah, like what's a typical amount of vitamin C a rat gets or a mouse gets? What's like too much? What's the lethal dose of vitamin C? What else might you have to know besides that? Tail?
Where should you put it?
Where? Yeah. How could you what form does vitamin C come in? Is it a liquid? Is it? How should I get it? How should I give it to the mice? Talia?
How much the mice weigh?
Yeah. What is like the starting weight of the mice? What kind of cage do I keep them in? What type of bedding material? What are different types of mice? There's lots of things we want to know ahead of time. Okay? So there's a whole host of things we might try to figure out before we set up our experiment. All right, then we can make our hypothesis. what would your hypothesis be? In this experiment, so the question we're asking, do mice gain weight if they are given extra vitamin C? Who could tell me what your hypothesis would be? David?
Yes, they do gain weight.
Okay. Can we phrase that as an if-then statement? Go ahead, Kenny.
If the mice are given too much vitamin C, then they will gain weight.
Okay, so yeah, if mice are given, let's say, extra vitamin C, then they will gain more weight, probably than mice that aren't. That's a hypothesis. If mice are given extra vitamin C, then they will weigh more. Could we test that in an experiment? Yeah. How would we do this? Anyone like envisioning this in your head? Like how would I set, if I want to set this experiment, I want to test that hypothesis in class. What exactly would I have to do? Richard?
You have to get a mouse.
Okay. Get a mouse? That's all?
Well, then you would have to get vitamin C
Okay.
and then feed it to them.
Okay. And?
See if it gains weight.
Okay. Good experiment. So I have one mouse, got a little cage over here. I give it water and food, and I give it vitamin C, and then I weigh it. Is that going to test my hypothesis?
No.
You have a problem. Emily, what's the problem?
That you don't have another mouse to compare it to?
Yeah, I'm not sure. So if I say, all right, this mouse weighs 532 grams at the end of the experiment, did it gain weight because of the vitamin C? I don't know. So what Emily said is important. How would you change it, Emily?
You would get another mouse and not get extra vitamin C?
Okay, so now I need two mice. One mouse gets just normal food and water. The other mouse gets normal food and water plus what? Extra vitamin C. Is that a better experiment?
Yes.
Yes. Having one of each mouse, how can I make that even better though? Because what if this is some weirdo mouse that just happens to be like really skinny? What would I do differently?
Having them like at the same health level
Okay.
for the experiment.
They'd have to be the same sort of health. What else?
Like make them do the same weight before you
Same
start?
starting weight. So I'd have to make sure there are other variables are controlled. And
Have a couple more mice just so you have multiple tests.
yeah, because one of these mice just happen to be like smaller or slower growing. So if I have three, what would be better than one mouse? Three. What would be better than three?
4,
What would be better than four?
5.
Okay, so you get the point. If I had 1,000 of each mice, then I could really be sure of my results. It'd kind of be a mess. But the more, we call those samples. The more samples you have, the better your experiment. What we would say is the more valid your experiment, the more sure you can be of the results. So, yes, Emily was correct. I would need two groups of mice. One group, I would feed normal food and water and vitamin C. The other group, I would feed the same, but they would get twice as much vitamin C. And that's going to help me to actually test my hypothesis. We got 2 minutes. What would I observe about the mice as my experiment is progressing? What are the observations I would make? Jeremy?
If it's getting bigger,
So what would I measure?
its size or weight.
The weight of the mice after a couple weeks. Let's say I do that. The vitamin C eating mice weigh the same as the non-vitamin C eating mice. Those are my observations. And your observations are the data you collected. So what would I conclude? So your conclusion, you say, was my hypothesis correct or incorrect? So based on this experiment and the observations, Christopher, what would a proper conclusion to?
My hypothesis was not meet that.
Was incorrect. So do you erase it and change your hypothesis to match what your data showed? No, you don't change your hypothesis. It was not correct. That's okay. Have we still learned something?
Yeah.
Yeah, we learned that giving mice additional vitamin C does not cause them to grow larger. So we still have learned valuable information, even though our hypothesis was rejected. All right, any questions? All right, have a wonderful day, students.
You too.
Well, you guys are today's